Welcome to this tutorial video. So in today's session, we just try to answer some questions that were, released in your tutorial sheet, biochemistry tutorial sheet, and specifically in this video, we'll look at the questions that are on amino acids metabolism. So before we even begin, I would just like you to understand certain things in general before I start talking about the actual questions that were put in this particular document. So, when you are talking about amino acids metabolism, what you must understand is that unlike what you've been learning to say glucose can be stored into glycogen, excess fats can be stored in form of triglycerides, all right? These particular amino acids, the body does not store them in any form at all. They are not stored. So they are always performing certain specific functions. For example, you find that maybe these amino acids are being used to form proteins that must form specific enzymes, right? Maybe they are being used to form proteins that must form specific neurotransmitters or specific structures, or be used in the production of different tissues or cells. So the body does not store these particular amino acids, but it keeps them in use. What does it tell you? If the body does not store these amino acids, it simply means that it must regulate how much is coming in and how much must go out. That's why you've got this concept of what? Amino acids metabolism. So this is a process that enables the body to balance these amino acids. What it must have inside and what it must excrete. Because if it cannot store them, it means because you keep on eating, for example, there is food that you eat that contains proteins, of course, and those proteins are made up of specific amino acids. So you just want to keep on eating those particular foodstuffs without it degrading these amino acids. So those that you don't want, the body will be able to get rid of them through what we call amino acids metabolism. Then those that you want, of course, the body will be able to form those specific amino acids through the same amino acids metabolism. So that's basically the whole idea that is behind amino acid metabolism. Simply, the body forms amino acids that the body wants from different amino acids, and it's going to get rid of those that it doesn't want, those that are in excess, through amino acids metabolism. So I know that you've read about these things. You've got general understanding about these particular things. Let's just see how we can answer questions on this particular topic. Now, the first question reads, so organisms vary in their ability to metabolize and synthesize amino acids. That is very true. That is question one, saying organisms vary in their ability to metabolize and synthesize amino acids. Question A, distinguish between point number one, or rather what we call I. So distinguish between essential and non-essential amino acids. So the first question we're answering is the difference between essential and non-essential amino acid. So this is a very simple question because when you're talking about essential amino acids, you are talking about amino acid that the body cannot synthesize on its own. So these amino acids must be what? Must be obtained through diet. So amino acid that the body cannot synthesize on its own are what we call essential amino acids. Of course, there are different types of these amino acids that you may come across, all right? You can check out those. But what I just need you to understand is that the essential amino acids are those that the body cannot synthesize biochemically. So they must be obtained through diet. And then when talking about the non-essential amino acids, you are talking about amino acids that the body can synthesize because they're non-essential. You must understand that when you're talking about something essential in biochemistry, you're talking about something that the body cannot synthesize. That's why we're saying that the essential amino acids are those types of amino acids that the body cannot synthesize on its own. Then if you're talking about the non-essential amino acids, these are types of amino acid that the body is able to form on its own throughout this amino acid metabolism. For example, you can talk about glutamate, alanine, glutamine. Those are types of these non-essential amino acids that the body can form on its own. So that is the key. The essential ones, the body cannot form them. The non-essential ones, the body can form them. So you can just download pictures of the specific types because there are a lot. Okay, there are a lot. of these, essential amino acids, and there are a lot also on the non-essential amino acids. You can look at those particular, examples. So away from that, let's also look at this question, which is saying we're supposed to distinguish between ketogenic and glucogenic amino acids. So before I even go into talking about this particular question, you must understand that amino acids are classified in different ways. For example, you've heard about amino acids being classified in terms of being essential and non-essential, like we're just from mentioning. Then I'm sure you've also heard about amino acids being classified as polar or non-polar, positively charged or negatively charged, okay, aromatic and non-aromatic. You've heard those examples. But another way in which you can classify amino acids is when you use the metabolic activity. So it's like, what kind of metabolic process can this amino acid undergo? So we've got what we call glucogenic and ketogenic amino acids. So that can also be used as a classification. So when you're talking about ketogenic amino acids, just from the word keto, it simply means that these amino acids can be used to form what we call ketones or ketone bodies. I've got a video on ketogenesis. I'm sure you've seen how those ketone bodies perform specific functions in the human body. So when you're talking about ketogenic amino acids, we're talking about amino acids that are used in the synthesis of what? Ketones, or what we call ketone bodies. That is the general picture that you must understand when you're talking about these particular. For example, you've got lysine, I think you've got leucine amino acids that fall under the what? The ketogenic amino acids. Okay? Then, talking about the glucogenic amino acids, just from the term glucogenic, you can see that this term is trying to describe amino acids that can be used to form what? Glucose. So, glucogenic amino acids are those amino acids that can be used to form glucose. How can they do that? It's after they undergo this amino acid metabolism. Okay? So, for example, you can talk about alanine, you can talk about glutamate, glutamine. Those are examples of glucogenic amino acids because from them you can form molecules or carbon skeletons that will be able to form glucose. So basically, ketogenic amino acids are those amino acids that can be used to form glucose. Ketones or ketone bodies. Then glucogenic amino acids are those that can be used to form glucose. That is that. Now, talking about the third question, which is just saying that we need to differentiate between transamination and what we call racemization. Okay, so the difference between transamination and racemization is that you find that when you're talking about this amino acid metabolism, when talking about this amino acid metabolism, it happens in different ways. One of the ways is what we call transmination. One of the ways is what we call oxidative deamination. One of the ways is what we call transdeamination. Okay, so basically, when we're talking about transamination, you're just talking about, just from the word trans, it's just talking about transferring. What are we transferring? Okay, you can see that transamine is coming from the word we're transferring an amino group, hence the term transamination. So basically, transamination, this is a metabolic process in which you transfer an amino group from one amino acid to another amino acid. But if you had to ask yourself, why are you doing this transferring process? It's simply because, for example, if your body needs alanine, right, as an amino acid, and then what you have is glutamine, for example. So the body is going to say, okay, what I need is alanine. So there's what we call a keto acid. So a keto acid is going to be available, but it's going to be incomplete. Why? It's because it doesn't have an amino group. Remember, each amino acid, for it to be complete, it must have that amino group, that carboxyl group, that side chain, and that hydrogen there. Now, what the body does to be able to form new amino acids, it is going to, for example, I said if you need alanine, so it's going to get an amino group from glutamine, for example, from glutamate, for example, and give it to a specific alpha keto acid that can be used to form alanine. Then the body will successfully what? Form that alanine. So basically, when talking about transamination, we're just talking about the transfer of amino group from one amino acid to an alpha keto acid to form a new amino acid and a new alpha keto acid. Because why am I saying you're forming a new alpha keto acid? Whenever you transfer that amino group from one amino acid, it's going to remain without what? That amino group. So if it doesn't have an amino group, it becomes what you call an alpha keto acid. When you get that amino group, you give it to another or different alpha keto acid. Of course, by the virtue of you giving it that amino acid, it's going to become a new amino acid. So that is what is all about, when talking about transamination. What I would just like also to mention is that this process of transamination takes place where it takes place in the cytoplasm of the cell. Okay. Okay. Now coming back to the other term. So coming back to this other particular. Okay, let me just. Okay. Okay. I hope that is still visible. So coming back to the difference between transamination and racemization. So when talking about racemization, this process simply talks about converting what we call an L amino acids into a D amino acids. In other books or other contexts, you find that they like referring to them as it's a process that involves converting a mirror image of another amino acid. For example, I said this process of racemization is whereby you are converting, for example, you've got, I'm sure in most of you that did natural sciences, you've heard of things like L-glucose, D-glucose, okay? Those are just the different structural presentation of, if you are to look at those particular molecules in the 3 dimension, they would appear as L or D glucose. So it's the same concept that we're using. So, for example, if we're talking about racemization, we're trying just talking about how you can convert an L amino acid into a D amino acid, just like a mirror. If you're looking at yourself in the mirror, I'm sure you see that there's that visual representation. Your right is going to be on your left because you're looking yourself at a mirror. Okay, so basically this racemization process, it is used in forming different dimensional structures of these amino acids. For example, you can move from an L amino acid to a D amino acid, and this particular process usually takes place in bacteria when they want to form different types of amino acids that they want to form. Of course, they're going to use this process of what racemization by converting L amino acids and converting them to D amino acids. So the main difference is that transamination only involves the transferring of an amino group from one amino acid to the alpha keto acid to form new amino acid and a new alpha keto acid. While racemization, this is a process that just involves the conversion of an L amino acid into a D amino acid. That is basically the difference that is there. All right. So having looked at those particular questions on A, let's move on and look at question B. So question B says, why are transamination reactions vital in both synthesis and degradation of amino acids? So if you got me very, very well, this question is very simple because I gave an introduction that what your body does when you're talking about amino acid metabolism, you're talking about how amino acids can be formed or degraded. Not formed from the coding when we're talking about translation, transcription, translation, no. We're talking about how the body can use the already available amino acid to form another one. So remember I told you, the key concepts about this particular amino acid metabolism is you understanding what we call an alpha keto acid and an amino acid. So an amino acid, of course, this is the building block of what? Of proteins, right? Then what is this? What is an alpha keto acid? An alpha keto acid is simply an incomplete, I'll put it in this term for you to understand. It's an incomplete amino acid without the amino group. It has everything, but the only problem is that that alpha keto acid has no amino group. So immediately to just give it an amino group, you're going to form a new amino acid from that alpha keto acid. So what is, kind of coming back to the question, what is the importance of this transamination process? Remember I said the first option that you can give is that it is used in balancing the levels of amino acids in the body because you wouldn't want to have excess types of amino acids in your body. Okay, so you'd want everything to be in equilibrium. So how do you do it? For example, if you've got too much glutamate, you've got too much glutamate in your body, and then your body will be like, I think this glutamate is too much. It can cause maybe complications, especially in the brain. So what the body it want to do is that it want to reduce those levels of what glutamate. What is it going to do with it? It can perform this transamination. So it's going to be getting those amino groups on that glutamate and giving it to other alpha keto acids to form different amino acids that are what that are minimal. Okay, so transamination is used in the regulation of amino acids amount in the body. That is that's one of the response that you can give. Apart from that, remember I said that, for example, if your body needs alanine and then what you have is glutamate or what you have is aspartate, but what your body wants is alanine. So your body can use transamination. How? It's going to get an amino group from glutamate and give it to an alpha keto acid that can form alanine. Then the body will be able to what? To synthesize that particular alanine that it what needs. That's why these transformation processes are, what, are very important. Apart from that, remember, as you are performing transamination, you're just not removing that amino group and giving it to another alpha keto acid to form a new amino acid. Remember, whenever you remove that amino group from that specific amino acid, it's no longer an amino acid. It has been degraded to what? To an alpha keto acid. So the body can also, this transamination can also be used to what? To degrade or break down the amino acids that are in excess. Those that you don't want too much. So just be like, okay, I think these are too much. Let me remove these amino groups. Let me give them to other alpha keto acids that can be used to form other amino acids that are not in excess, of course. So transamination is important in regulating the amount of amino acids in the body. Apart from that, it is used in the formation of amino acids that the body wants and in the degradation of amino acids that the body does not want. So that is the response on that particular question. Apart from that, we can also look at question C. So they are saying that name 7 metabolic products that arise from the degradation of what? Amino acids. So there are different products that the body can produce after degrading these what? Amino acids. So if you've got background information, for example, you've heard about the urea cycle, or maybe you've just heard about this topic in general, talking about amino acid metabolism. Remember, we talked about amino acids being degraded or being formed. So one of the products that is formed, I know that most of you are familiar with this, is what we call ammonia. Remember, after an amino acid is degraded, whether you use transamination or transamination, okay, or oxidative deamination, they will of course be in all the pathways that I've mentioned. There will just be that removal of that amino group, no matter what. So that amino group, of course, can be used to form what? Ammonia. And that ammonia, of course, you don't need it in your body because it is toxic. Okay, it can, cause a lot of complications if it is too much in the body. So one of the products is what? Ammonia. So apart from ammonia, the body can also form urea from what? Amino acid metabolism. Remember, when you form ammonia, that ammonia is used what? Is used in the urea cycle to form this urea. Apart from that, carbon dioxide can be produced from this amino acid metabolism. Water can be produced. Ketones, remember we said they are what we call ketogenic and glucogenic amino acids. So for example, if you've got this amino acid, you break it down, you're going to form ketone bodies or molecules that can be used to form ketones. So ketones can also arise from this metabolism of what? Amino acid. Apart from that, glucose. Remember we said we've got what we call glucogenic amino acids. So if you have to break down these amino acids, if you have to degrade them, you can form different structure that can be used to form glucose. For example, you can form pyruvate from alanine. Of course, that pyruvate can of course undergo gluconeogenesis and it can be used to form what? Glucose. So apart from that, ATP molecules can also be produced. So in general, if I were to mention the 7 we've talked about, actually there are more than 7 that I can actually mention. We've talked about ammonia, we've talked about urea, carbon dioxide, water, ketones, glucose. If you want to go in details, you can talk about neurotransmitters. For example, these amino acids can, if at all if you're talking about the synthesis, they can be used to form dopamine, serotonin, different neurotransmitters. So you can have ammonia, urea, carbon dioxide, water, glucose, dopamine, serotonin, these ATP molecules, and many other products that can come from this amino acid metabolism. So that is what I just wanted you to know on this particular question C. Apart from that, they are saying that D, why are the amino acids glutamine and glutamate central to amino acid metabolism? What is the function of this ATP in the conversion of glutamates to glutamine? So I'll start with the first part. Why are the amino acids glutamine and glutamate central to amino acids metabolism? So for those students that may be struggling with the difference between the two, what is the difference between glutamate and glutamine? So I'll start with what glutamate for you to understand. So glutamate, we actually say this is the most common amino acid that is abundant in the human body, glutamate, because it is used in what we call excitatory processes in brain mechanism, especially if you're talking about the nervous system. Okay, it is used in causing excitations of neurons. Okay, so you find that it is too much. Okay, it's in abundance compared to other amino acids. I'm not saying too much because it's toxic. I'm saying it's the one that is in abundance compared to other amino acids in the body. So what happens is that when you've got this glutamate, this amino acid, this glutamate, When you combine it with ammonia, you form glutamine. When you've got the glutamate plus ammonia, you form what? Glutamine. Now, the reason why we are saying that these amino acids are central to amino acid metabolism is because since you've got too much glutamate, you can use that glutamate to form other amino acids that, like I've mentioned, from transamination processes, from oxidative deamination, from transamination. You can use this glutamate because if you've got too much of that, you can just be getting their amino groups and give them to other alpha keto acids that can be used to form what? Other new amino acids. So you find that this glutamate becomes more like a central point of which the body can use this amino acid metabolism mechanism to be able to form other new amino acids that it wants. If it wants alanine, if it wants aspartate, if it wants glutamine, just like that. So you find that this glutamate becomes a central point, and you must understand that this is very, very important, the glutamate, amino acid. Now, talking about glutamine, remember I said glutamine is just a product that comes from glutamate. What you do is that when you combine glutamate plus ammonia, you form what we call glutamine. So, what is the importance of this glutamine? Remember, ammonia is not good for the body. You don't want ammonia in the body. No wonder we make that ammonia to undergo the what? The urea cycle. I'm going to show you very soon. Because so that we can convert it to something that at least that the body can manage, which is just urea. Okay? That you find in urine, of course. So this glutamine is used to reduce the toxic amount or the toxic levels of what? Of ammonia in the blood. So instead of ammonia just moving on its own, okay, as ammonia, what you do is that you get glutamine, you get glutamate, you combine it with what? Ammonia, then you form what? Glutamine. Then you're going to hide that ammonia inside glutamine because glutamine is just a combination of what? Ammonia, is a combination of what? Ammonia and glutamate. So this glutamine now is going to be helping to regulate those excess levels of what? Ammonia in the what? In the blood. Because if you've got this ammonia that's just moving on its own, then that is going to be causing a lot of damages on organs, on different, biochemical pathways. So what the body does is that if it sees to say, okay, I've got excess ammonia right now, what can I do? Let me get glutamate and combine it with this ammonia. What I reduce the toxicity of what? This ammonia. Then you'll be able to form this what? Glutamine. That's why I find that this glutamine and this glutamate are more like the central points of this amino acid what? Metabolism, because one, they regulate the levels of what? Ammonia in your blood. Apart from that, glutamate, we've said, it's more like the most abundant amino acid that a human being has. And from this glutamate, you can use it to be able to synthesize other what? Different types of what? Amino acids. Okay? What is the function of ATP in conversion of what? Glutamate to glutamine? Very, very simple question. Remember I said, when you're forming glutamine, you're just combining glutamate plus ammonia. Okay? Glutamine plus what? This ammonium. And then after you do that, remember in that particular pathway, as you are combining glutamate and ammonium, what you are doing is that you need energy to be able to drive that reaction. So what are you going to do? You just need ATP to drive that reaction. As you are breaking those particular bonds on this ATP molecule through the hydrolysis pathway, you're going to release energy, and that energy is the one that is needed to combine that ammonia plus that glutamate to form what? Glutamine. So that's basically what this part is trying to ask us. So I hope that has been noted. Question E. Why do marathon runners prefer beverages with sugar content instead of amino acids during long race? Explain why ketogenic amino acids yield. Explain why ketogenic amino acids yielding acetyl A or only cannot be converted into glucose. Okay, very, very good. It's a very wonderful question. I like this question very, very much. So I'm going to answer it in parts depending on how it has been phrased. So I'll start with the first part. Why do marathon runners prefer beverages with sugar content instead of amino acids during long races? So for example, you've heard when you go for sports, you would want to drink, for example, glucose, okay? Or they will give you foodstuffs that contain too much glucose. Remember, before I even talk about the amino acids themselves, I'll talk about glucose itself first. Remember, glucose can readily provide the much needed ATP through glycolysis in about just 5 minutes. Okay, you are going to have this particular ATP readily available from glucose, because from glucose you just undergo glycolysis straight and you form ATP. And remember, from glycolysis you can form pyruvate. pyruvate will directly form acetyl-CoA, which can undergo the Krebs cycle, produce NADH, FADH2, and those are used in neutron transport chain to be able to form ATP. So glucose will provide the much needed energy in a very short period of time because glycolysis is very fast. Now, why do you think marathon runners would not want to eat would prefer sugar content food instead of amino acids? Remember, from amino acids, what will happen is that the first thing you're supposed to do, you're supposed to degrade these amino acids. Remember, we said we've got glucogenic and ketogenic amino acids. So you must look for these amino acids first that can undergo, that can be used to form glucose. For example, you're supposed to find alanine, okay? After you find alanine, you break it down to what? To pyruvate. Now, from pyruvate now, you must now find a way, okay, of making that pyruvate to be able to leave the mitochondria, to be able to perform what we call gluconeogenesis and form glucose back. After you form glucose, that's when you start making that glucose to undergo glycolysis. So it's a very complicated process. Why? Because it's a long pathway. Imagine you've got amino acids, of course, that are there, but for you to form glucose, you must break them down. From that alanine, you remove the amino group in transamination, then you form pyruvate. After you form pyruvate, you make pyruvate to be able to undergo gluconeogenesis so that you can form glucose. After you form glucose now, that glucose now is now used to form what? To form, it's now used to be able to perform glycolysis again. It's more like just going back again to pyruvate. So it's more like a long process because marathon runners, they will need ATP in availability, in readiness. They wouldn't want to start wasting time in doing transamination from amino acids, then from there gluconeogenesis, then from there again back to glucose, back to glycolysis. That would be very complicated. That's why they'll just prefer to eat foodstuffs that contain readily available sugar that can undergo glycolysis right there and then. Now, explain the other part. Explain why ketogenic amino acids yield acetyl-CoA. Let me just rephrase this question again. Explain why ketogenic amino acids yielding acetyl-CoA only cannot be converted into glucose. Remember, we said amino acids have got ketogenic and glucogenic. Talking about ketogenic, there are those type of amino acids that can be used to form what? Ketone bodies. For example, ketone bodies, we've got acetone, acetoacetate, you've got beta-hydroxybutyrate. Now, the problem is that when you form those ketone bodies from these amino acids, the problem is that those ketone bodies, they can only form acetyl-CoA. And acetyl-CoA cannot be used to form glucose directly. No, it must be diverted into other different molecules again that must find themselves in performing gluconeogenesis because acetyl-CoA cannot be converted into glucose directly. Yes, it can be used to as a precursor of the Krebs cycle, part of the Krebs cycle, but it's not used to form what? Glucose directly. So they would definitely just prefer something that has got glucose already, because even if you obtain these ketone bodies from these amino acids, they'll just form acetyl-CoA, which is not glucose. And it's like I've explained, it's difficult or rather complex, sorry, or rather a complex pathway for you to be able to move from that acetyl until you form glucose. It's a very long pathway. So marathon runners would not prefer such kind of situations. They wouldn't want to form these ketones. They wouldn't want to use these amino acids. They would prefer just using glucose directly. I think that question has been what? Answered. Okay. Now, coming to this particular question, which is just question F. So alkapotnuria, okay, is a condition where urine turns black. Biochemically explain the root cause of this disease. Okay, alkapotnuria is a condition whereby urine turns black. Biochemically, Biochemically explain the root cause of this disease. Okay, apart from that, what specific amino acids are involved in alkapotnuria? So it's a very simple question. It's a very, very simple question. So, when talking about this alkapotnuria, of course, you are talking about a condition that makes urine to turn black. So, you find that somebody is urinating, but the type of urine that they are producing is black. It's because there are amino acids that are in that urine that are too much or that was supposed to be what broken down instead of them being,